So I would like to welcome everyone to our first talk of these e-seminar series on translational biomedical engineering. These series are aimed to serve as a venue for academic researchers, members of industry, clinicians, regulatory officials, patent experts, entrepreneurs, and investors to discuss advances and challenges in the development and translational biomedical technologies. So before I start, I have a few housekeeping points that I wanted to share with you. This talk will be recorded and will be posted on our YouTube channel. We will send you a link after this meeting in a follow-up email. If you have any questions during the talk, please use the raise hand button or write your question in the question box. We will unmute you and then you can ask your questions from our speaker. We also have a survey for these series. A link to a survey will be shared with you after this talk. Please make sure that you participate in the survey and provide your feedback to us. We need your feedback to improve the quality of this initiative. I also would like to thank Revolution for sponsoring this talk and supporting our initiative. So let's go to our first speaker of this seminar series. I have a lot to talk about Ali. He is my ex-mentor and a role model. Ali is basically, he is the CEO of the Terasaki Institute for Biomedical Innovations, and he is the co-founder and the scientific board member of Obsidio. Before starting the new position, Ali was the Levi Knight Professor of Bioengineering, Chemical Engineering and Radiology, and the founding director of Center for Minimally Invasive Therapeutics at UCLA. Before joining UCLA, and when I had a chance to work with him, he was a full professor at Harvard Medical School and a faculty at Harvard MIT Division of Health Sciences and Technology, Brigham and Women's Hospital, and he was also an associate faculty at the Wyss Institute for Biologically Inspired Engineering. Now things get more exciting. Ali has edited multiple books, and he is an author on more than 750 peer-reviewed journal articles, book chapters, review papers, patent disclosure applications. He has been cited, oh my God, 65,000 times, and he has an H index of 131. So annually for the past 5 years, he has been selected by Thomson Reuters as one of the world's most influential minds as a highly cited researchers. I'm not done yet with Ali, so it takes a while to go over Ali's credential. So Ali's interdisciplinary research has been recognized by over 60 major national and international awards. So he's only 5 years older than me, and then the number of awards that he has received so far is the same as the number of papers that I have published so far. So it's almost the same. So you can see who my mentor is. So his notable awards are the Presidential Early Career Award for Scientists and Engineers, the highest honor given by the US government for early career researchers. Barack Obama, I think, gave you Ali this event, and you get to meet him. He also received the Mustafa Prize, $500,000, based on your website, Ali. I mean, this is a very prestigious award, and the Clemson Award of the Society for Biomaterials. In 2007, he was named a TR35 recipient by the MIT Technology Review magazine as one of the world's top young innovators. I can go on and on about Ali, but we have a limited time, and then I'm sure that everyone is very eager to hear Ali's thoughts on engineering and precision medicine. Welcome, Ali, to this virtual meeting, and the virtual floor is now yours. Let me just share change presentation, share the screen, so you can share your slides, and then we'll hear your talk.
All right. Super. Let me see what you guys are seeing. This is just a blank screen that you guys are seeing now? Or do you see my screen?
No, blank screen.
Oh, okay. Let me end share and then reshare. Unshare, stop sharing. Oops, that was my webcam. Now it's going to disconnect my audio. Degenerating. All right, share my webcam and share screen. Sharing, stop sharing screen, okay. And my screen here. Now you see something.
We see
right. Now you see this. Now you see this. All right. I need to not share my main screen. I need to share my presentation.
There's
which means, yeah. Good thing we only tested this like 5 times before. There we go. Hopefully this will. Awesome. All right. All right. Thank you, guys. Pleasure to join you. I'm super excited about your seminar series, and obviously looks like there's a lot of interest. I see many hundreds of people that are signed in, so congratulations to you. I was mentioning to them earlier that I need to know how they advertise their talks, because when I advertise my own talks, not as many people show up. So so it's also a great pleasure to see Mohsen and Human just do incredible things. I've seen them since they were yay high junior researchers, and now they're all professors. So it's amazing to see them develop. So today I'm going to give a little bit of a different talk because of the specific focus of this this seminar series, which is more on translation. And I thought maybe I give some of my perspectives about my own experience in entrepreneurship and translating research, and discuss some of the challenges that I had to go through and I'm still going through in doing this. So it's I think hopefully it will be educational. So as many of you guys heard, I've recently started to take a position at the Terasaki Institute. Terasaki Institute is a new initiative, and it's enabled by Dr. Terasaki, who is pictured here, who really developed one of the first personalized medicine applications. So what he developed built on this picture. This is the picture of the first transplant that happened at the place I used to work at Brigham Hospital. And in this picture, Dr. Joseph Murray performed the first kidney transplant between 2 identical twins. Now, with identical twins, you don't have to worry about mismatch of tissues, and it's not going to get rejected. But if you're dealing with just anyone giving an organ to someone else, there's a huge chance of mismatch and rejection. So what Dr. Terasaki developed was a technology to be able to identify who is the right donor for which recipient, and based on that, eliminate a lot of the issues that existed with transplant. Based on this technology, he started a company that then they sold a few years ago to Thermo Fisher, and based on some of the proceeds, he started to build a foundation of which the Institute is a big beneficiary. So the Institute, what we're trying to do is really build on the legacy that Dr. Terasaki started on innovating personalized wellness and cures, so really personalized health. And our mission is to invent and foster real solutions that enhances the health of individuals. So that's what we're trying to do. So when you see what our mission is not just doing research, but also fostering their applications, really pushing them through this process. And we're located in Los Angeles, which is a beautiful city. We have now 3 different buildings. These 2 buildings on the left-hand side are our legacy buildings, which are grandfathered from Dr. Terasaki, and we recently bought a new building, which is going to be our flagship building here in Los Angeles, which is going to be a bigger building, about 50,000 square feet, which is going to be added to our additional building. So overall, there's a significant amount of space that we're trying to build to really make a top-notch research institute here in Los Angeles. So I definitely encourage everyone to start interacting with us and looking at our website. It's been recently developed and is still up and running, but and still being further edited. But you can now start to look at some of the different pages, and you start getting a sense of some of the things that we're trying to do with personalized health. And when you think about this concept of personalized medicine, or more also referred to as precision medicine, what people have been trying to do is to try to understand the variability that exists between individuals, and based on that, be able to tailor medical treatments or diagnostic approaches. So at the Terasaki Institute, we're looking at this whole thing differently and in much more of a holistic approach. So typically what the whole medicine field is based on is the top approach, which involves a lot of omics, where what people try to do is basically try to sequence the genome, look at the proteomics, look at the glycomics, and do bioinformatics and similar kinds of things. And based on understanding how this molecules are expressed in the biological tissue, try to identify what kind of drugs, for example, would be beneficial for the patient. Now, this is what we think is a very powerful approach, but it's not a cohesive and fully integrated approach with a lot of other things that exist right now. So what we're trying to do is to really bring different aspects into this, not only understand the genetic and proteomic kind of information that is associated with an individual, but bring in technologies that have been developed by engineering, chemistry, and other types of sciences, so we can actually have a much more cohesive approach in addressing each individual's medical needs. And the way we do that is we've built a series of platforms that involve things like personalized devices that can monitor and treat individuals. These are based on basically advances in electronics and other types of sensing capability, wearables, so we can actually understand what each person needs. Alternatively, we're also interested in developing personalized implants and regenerative medicine approaches, and this is again based on things like understanding what the individual needs through non-invasive imaging and biological markers that they have, and then be able to design 3D print perhaps specific constructs that will be relevant for that individual. Also, we have a couple other platforms, personalized models, as well as personalized materials, which are again aiming to develop new technologies and be able to make models of specific person's physiology so we can understand their disease and then be able to address it. So this is what we're working on at the Institute, and I encourage people to connect with us. So one of the things that I think you should all do right now, many hundreds of you, is to connect with us through our social media platforms, both the Institute platforms as well as my own personal platforms, where I will be very happy to give you a lot more information about these types of things that we're doing. And also, I encourage people to go to our website and look at some of the other things that we have. I've putting a shameless plug for some of the things I'm doing. I'm also doing a seminar series as well as a series of fireside chats on various topics, which I think people will be interested on. And also, I'm interested in hiring the best and the brightest at the Institute. So we are hiring a number of positions at various levels, and we're hoping that people who are interested in this field can join us. So let's get started with what the topic today is about, which is really on how do we try to make an impact with the kind of research that we're doing. And I couldn't start the talk in any other way than to talk about Bob Langer. Bob Langer was my PhD advisor when I was getting my PhD at MIT, and as many of you know, he is really the pioneer scientist and the person who's been many times basically compared to Edison in his development of real-world practical applications that has been able to launch dozens of companies and over 1,500 patent applications. So before we talk about my own experiences, I wanted to tell you what I learned from Bob about how he goes about doing innovation and how he goes about starting companies that he has developed. And I think that's important to know because at least it gives you a perspective on what is the approach that has worked over and over. So these Bob's rules basically are as follows. There's 5 of them. You know, try to develop a product company that has a strong product that can be sold in the market, and it's got a big market ideally that people will have interest in. Try to really make something that is a platform technology. So try to really do something that is not just a one-trick pony, but is something that can be applied to many different applications. So if you have a technology that can be used for diabetes and at the same time can be used for cardiovascular disease and musculoskeletal disease, then that's the kind of stuff. It's a platform approach that can be used for many things. And just to give you an example of that, Moderna Therapeutics, which is one of Bob Langer's recent companies that has gained a lot of attention, started off as a company that was making RNA-based drugs for different applications. But when COVID situation came earlier this year, Moderna started to pivot into building or developing COVID-19 vaccines. And that just shows how much of a platform technology it is. It's actually allows them to pivot into different areas because the technology is adaptable to different important needs. The other thing is to really show that it's scientifically strong. So look at seminar article and really try to publish a groundbreaking paper in a leading journal, and that's another part of this. And finally, to try to not only publish a great scientific paper, but also be able to create intellectual property and ideally a blocking seminal patent or family of patents that can protect the position of the company, the new company that's getting launched. And this is very important. Again, publication is very important to get the science out to the real world, but without having intellectual property protection, it's hard to get the thing launched because investors are not going to be as vested or interested in trying to launch the company because then anyone else can copy the technology. So actually the patents winds up enhancing innovation as opposed to decreasing it. And finally, this concept of don't do it too early, develop the technology, push it as far as possible, within the laboratory before you actually launch the company. And in many of the types of cases that are biomedical, having the proof of principle in animal models, which are basically the preclinical models to human trials, is very important, so that it's all been validated, vetted, and the startup can now raise the funds to be able to do the human clinical trials. So some of the other things that I would say are important is to really focus on this high impact problems. You know, and I've learned that through experience that you can develop something that's really interesting and differentiated and has a intellectual property that's protected, but unless it's a big market, affects a lot of people, investors are not going to be as excited about it. And that's really important for really bridging this valley of death, this what they call the barrier between what we do in a laboratory at universities or research institutes and what is needed to make that innovation come to real world. And bridging this valley of death really does require understanding not just the science, but also what is the market, investors, customers need to be able to do that. And one of the things that, again, these last 2 points about really building collaborations and creating convergent ecosystems are also very important. So I think one of the things you may think about Bob Langer is that he is one person, a genius working and developing all these things. But in reality, that's not how it works. And I like this analogy here. So this is you know, Newton, a cartoon of him, and of course the apple you know, hitting his head, and then he's coming up with the idea of you know, really this whole F equals ma, and revolutionizing physics at the time. Now this is how maybe science and really big breakthroughs were done hundreds of years ago, but this is no longer the case. Problems are much more complex and require many different fields to join together and be able to address them. So this is much more like it now, where Apple has to hit the head of many people before the idea gets fully developed. And this is true also for Bob. Bob doesn't work in vacuum. He works very closely with many other stakeholders. In fact, when I entered science, his innovations in working with a clinician, particularly Jay Vacanti, who is a surgeon at Mass General Hospital, developed this whole field of tissue engineering, which got me very excited when I was finishing my undergraduate to enter this field. And it's been just continued for him. He's developed a lot of different teams with different people to be able to deal with different problems. And that's very important, creating a convergent science approach where there's very little barriers between people from different fields. And I wanted to give you an example of the kind of work that we do and how it involves different people. So this is actually a picture from one of the meetings that we've set up in trying to address particular problems. And in these meetings, what we have is that, as you can see, we have clinicians, engineers, chemists, biologists, and many different disciplines represented. And almost looks like Noah's Ark, because you have, you know, one of everything, in this case, not a pair, but at least one of everything, this different fields represented. And that's what it takes a lot of times to be able to understand the problem, as well as come up with innovative solutions about it. And in our own group, we also have a very similar mentality, where we do have people from all over working together, and people think differently because of their own scientific and personal backgrounds, and that actually allows them to come up with things that are much more than some of the individuals. And I've been also very, very lucky through the connections that I've made in science. So one of the amazing things about what we do is the interactions that we have as colleagues. Again, people from different backgrounds and different knowledge, expertise, working together. And I've been very lucky to have amazing mentors. Many of them are listed here and shown here. This is one of my favorite pictures, which has Bob Langer, who was my PhD mentor, as well as Michael Sefton, who was involved in my earlier training at University of Toronto, as well as Dr. Nicholas Pepys, one of the leading figures in biomedical engineering, and one of his students, Dr. Tony Miko. So it's just, I think it's great to have people who care about real world problems working and being able to interact with each other as well. So my own story that I wanted to talk about today is something that's, again, brought me in contact with a clinician to try to build something that would address his own clinical need. So this is Rami Oklu. Rami and I have become friends over the past few years, 5-6 years. And what Rami does, he's an interventional radiologist. So what he does is that he plays with catheters. You know, I often call him, he's the plumber of the body. He takes little tubes and he navigates blood vessels. He moves them around inside the body, and he can perform pretty incredible plumbing work, which is opening blood vessels or closing blood vessels based on the needs of the patient. So Rami and I met when I was in Boston, and what he presented to me was a number of problems that they've had, which involve things related to plugging blood vessels where you don't want, let's say, unwanted bleeding, or when you have aneurysms where you need to block off. And Rami and I worked together over the past few years to develop a material that I'm going to talk about here. And it's been a very productive collaborations. We've worked on many publications, a number of patents, and this startup company that I'm going to talk about. And it's been just an incredible experience. And I think when it comes to having these kinds of interactions with a person from different field, the best way to do it is if you're actually friends, because it makes things a lot easier. Of course, it challenges your friendship at times, but if you're moving in the same direction, I think at the end it's going to be beneficial. So this technology that I'm going to talk about really started off when one of my postdocs, along with Bob Langer's postdoc, Akhilesh Kaharwar, came to the lab. He's now a professor at Texas A&M, and he really introduced us to these class of materials that are silicate-based nanoparticles that have very interesting rheological properties. Based on some of this work, we started to build up on a family of materials that we created some intellectual property for. We started working with Rami and published a seminal paper in Science Translational Medicine, and we got more intellectual property and a couple of major R01 grants that demonstrated its importance in the scientific field. And Obsidio was founded late in 2018, and over the past year and a half, 2 years, we've been working on developing a number of different advances to it, including a couple funding rounds as well as some SBIR grants. So what exactly is this thing? So the material that we've been developing is a combination of a couple of components. One is gelatin, which is very abundant in tissues and different medical devices, combined with laponite. And these laponite is a form of nanosilicate, which is very small particles that are disc-shaped, and they have also asymmetry in their charges. As you can see on the edges, they're positively charged, and in their flat sides, they're negatively charged. And based on these charge distributions, they can interact with themselves as well as with the polymers, and create a material that is shear thinning. That means that if you apply force to it, it actually flows. And when you remove the force, it stops flowing. So this allows you to have these toothpaste-like properties, so that you can deliver it through a needle or a catheter, but once it comes out, it maintains its shape. So I don't know if this video is going to work with sound.
No sound.
Okay. All right. So basically, what it talks about is that in embolization, it's basically a procedure that basically allows you to go inside the blood vessel and block it. And this can be used for tumors to be able to starve tumors from oxygen by blocking their blood supply, as well as other areas. So this is really what embolization is about. Now, embolization is used in many different applications. So tumors is one of them, where you can take tumors and basically block off their blood vessels. It's also used in the brain to be able to address things related to aneurysms in the brain, as well as other types of bleeding that occurs in the brain. And finally, it's used in peripheral vasculature. So for many different applications when you need to, again, plug blood vessels, fill them, and be able to deal with it. So this is really what embolization is about. And if you think about this process, what's typically done is as follows. So this is an example of an aneurysm. So what interventional radiologists do is that they go in with a catheter, and then they kind of fill this aneurysm with these wires. Now the wires have been used for a number of decades. And what they do is that they basically slow the flow of the blood inside the area, and it causes the blood to stop and form a blood clot. So that's what happens. And but it doesn't work all the time. So this is an example. You see here is the guide wire for this catheter, and you can see that through it, lots of different coils have been deposited. But in this case, actually, it didn't really work. All these coils together did not stop the bleeding. And this is the kind of stuff that happens. So here you see, again, lots of these coils. And then once you put contrast upstream, you see that it goes directly through this, as you can see by this contrast coming downstream from here. So this basically shows that it doesn't work in every case. And the reason it doesn't work, and in this case, it's a gastrointestinal bleeding that occurs, so you see the bleeding continue. The reason it doesn't occur is that many people actually have impaired ability to form blood clots. So if you're gone into an accident and you've lost a lot of blood already, you can't form blood clots as easily. If you've are under treatments for blood thinning, so people who are on warfarin, for example, they can't form blood clots as easily. So in all of these cases, these procedures cause very high complications rates. So based on this problem that Rami presented, we came up with this material, which is, as I mentioned, combination of these two components, and we came up with a family of different materials based on changing the ratio of these two different components. And we basically identified as nanoclay with NC with percentage of this nanoclay. So you can either change the ratio of these two components, or you can change the concentration of the whole solid component in the liquid. And based on that, you can start getting different types of materials. So these materials
Ali, we can't hear you.
Hello. Hi, guys. Am I still connected?
Now you're connected.
All right. Sorry about that.
I can't see your camera.
Oh, you can't see. Okay, so let me redo the sharing.
Sure.
Sorry about that. I was on a VPN for my day job, and it just kicked me off. So I will come back on.
This usually happens to me a lot when I'm at home.
That's right.
Yeah.
So 1 second. And all right, now I can share. Do you see it now, actually, or not yet?
Not yet.
Okay. So stop sharing and
There you go.
show.
Awesome.
Well, that's me. Hold on. Show entrepreneurship. Okay. And boom. Wonderful. Now it's, oh, white screen?
No, it's your previous slide, but it's frozen.
All right. So let me. All right, show again. And oh. All right, so you guys don't see this.
No.
All right. But you do see this.
We can see you, but this slide is frozen. It does, I mean, this, oh, there you go. Okay, all right. So we see your PowerPoint, but if you,
Yeah.
right.
But when I make it into a main screen, you just see blank.
Yeah.
All right. So maybe I can just do it the other way. Do it the normal way, like this, if that's okay with everybody.
Yeah, I think that should be fine.
All right. So one of the interesting things about this whole thing is that this family of materials basically allows us to basically have properties that are shear thinning, but also ranges greatly in what the properties are. So if you make these materials from 9% and you start changing the ratio of these two things, you basically can have a lot of variability in amount of force needed to make them into make them flow. So you can get materials that are very strong and you can get materials that are very weak. And all of these materials basically have the ability to, once you put them under the force, they become liquid-like, and once you remove the force, they go back to their original property. So this is the kind of stuff that we actually want with the material, because now you can actually inject it, and once it goes in its place, then it forms the right kind of features. Now, we've done a lot of biological analysis and showing that it induces blood clot formation. So this example shows that what you can do is that you can have this material, you can have it in a way that it actually forms blood clots. So this is basically blood, a dish, and once you put blood, you see how long it takes to form the blood clot here. Typically, it takes a few minutes for blood clot to form. And even if you have gelatin without any nanoclay, it still takes a few minutes. Now, as you start adding this nanoclay, you get very rapid formation of the blood clot. So the time for blood clot formation with various concentrations of this material becomes much faster. The other thing that we can do is we can actually put it in different types of models, for example, seeing for its biocompatibility. So here, when we inject the material, we look at the biocompatibility over a few weeks. We see that the material doesn't have a huge amount of inflammatory behavior, and over time it degrades and it gets infiltrated with tissue. And this is a much more benign response than other types of hemostats that induce blood clot formation, like this commercial one, which you see even after 4 weeks has a lot of inflammatory response. We've done different types of animal models. We've taken rodents and seen if the material works in their blood vessels. Here you can see the material inside their blood vessel, and you can see initially there's a lot of blood flow. Once we put it in the femoral artery, we block the blood flow completely. And we've done a lot of large animal experiments. And this is, again, the kind of work that we had to do to make this material attractive. And this shows that in these large animal experiments, you can actually block big blood vessels. This is original, it flows, and then here you can see the flow has stopped. And this here you can see the tip of the catheter and the material. And the material basically works in a lot of different conditions that we've applied. So, okay, so unfortunately here, I have to do these one by one. But when you do other models, like when you, this is initially the entire tissue is open. Once you start putting in your material, you can start decreasing this blood flow completely, and you can actually, again, induce embolization. So we've done a lot of other studies. I don't want to bore you with a lot of them, but we've seen that the material is biocompatible. So if you put it inside the blood vessels remain blocked over long periods of time, and it basically, the material degrades and it's filled with infiltrating tissue. So it makes a fibrotic tissue response, which is the kind of response that we want long term. And many other experiments that shown its efficacy as well as its safety in different types of large animal models. So we did all this work, and then we wanted to actually translate this. Rami was super excited about this area, and he thought that it actually addressed one of his major clinical problems. So what we did is we started to think about forming a company which was aiming to redefine this therapeutic embolization area. So the first thing we had to do is to really find the right team. And a team that's really important, a team members that are really work well together are people who can communicate with each other, people who know their fields very well and are committed to the project. And we were very lucky to find Ehsan. Ehsan Jabarzadeh is also an academic entrepreneur. So he did his PhD in biomaterials. He understands the science, but also he did an MBA, so he understands the business side of it. So he partnered up with us, and he is very, very incredible person. I've been very, very lucky to team up with him. And Rami and I, of course, on the scientific side, were the engineer and clinician, and that's how we started off. Now the team has since grown significantly. We have a number of other advisors and other board members who are kind of involved now, but that's how we got started. And we actually had other starts with different team members, particularly different CEOs, which didn't work out as well. So that's really made me appreciate the importance of the right person who's going to champion the technology and push it forward. Really, all the work that Rami and I did was just a small part of what Ehsan has been trying to push forward. And now, over the past couple years, he's raised about $4 million in different types of seed rounds and different grants. And he's been really championing this, and it's just been wonderful to work with him. Now, what are the things that we had to learn through this process, which I think are going to be really important for everyone? So one is we had to really, beyond the science, dig into a lot of areas which were very uncomfortable initially for us. We had to understand the needs of the customers. In this case, the customers are the clinicians, but also their patients. They are all have a vested interest in the kinds of things that we do and what product is developed, and how we had to develop which market areas we're going to go over, because as I mentioned, this can be used in cancer, it can be used in embolization of aneurysms, it can be used in neuro applications. So there's a number of different things that one goes after. So we had to kind of define the opportunity and define the market. We had to look at the competitors, what other technologies were available, and what is their problems, what is the current standard of care, what do the hospitals like to use, what does it take for them to reimburse the process, who's going to pay for it. We had to look at our intellectual property portfolio and license these from the universities, and then the company has been building its own intellectual property portfolio as well. We had to look at a lot of things with regulatory, getting FDA approvals, the whole process of 510K versus a full pre-market approval, what they call PMA. and looking at the safety profile and the efficacy profile of our things. We had to do things that are even way beyond. We had to look at the production approach to make these in a GLP, GMP compatible process so that FDA actually accepts the kind of stuff that we do. We had to really dig a lot of things into investors and being able to really see what the different investors are excited about, who are the people in this space that are thing. And really, I think it's really important to kind of, you only have 1 or 2 shots on goal. So it's really important to go and pick the right thing to go after that gets investors excited, is an important problem, and is something that you can actually accomplish. So these were some things, what I can tell you about my own personal lessons is through all of this is the size of the market is very important, how well differentiated your technology is. This team, and particularly a superb CEO, is essential. And really a superb CEO can pivot, because what you start off with in your own head about what the idea is probably not what the company winds up doing. So it's important to really pick the right person who can understand opportunities and be able to pivot into the right areas. And of course, you need to be able to be ready to fail, and all of this takes a lot of risk that you have to take on. So so this is some general things, and I just want to maybe just for time's sake, I will just kind of tell you guys that we're trying to now institutionalize this process at the Institute, because I think that's one of the things that's very different in academia. It's difficult to get such things institutionalized. So at the Institute, what I'm trying to do is to try to create a pipeline where this process that I had to go through over a multi-year process and do a lot of learning through and development to understand it can actually be embedded into the fabric of the institution. So what we're trying to do is create a process in which we only work on transformative ideas. We have a huge vetting process that through which a lot of ideas come in, and only few of them are selected for us to work on. And this ideas should address very important problems, and the Institute will start to work with the investigators to de-risk the problems and cultivate them so that they can become viable startups. And really working on this calculated risks. If we're going to do big risks, let's make quick decisions, fail fast, move forward, and create a culture that values this industry academic partnership and allows us to really build things and focus on this translation. And also the culture, the type of people that are there are just as important. So being able to have the right scientists who value these kinds of work and be able to work with the best people in trying to make it happen. So that's really what the Terasaki Institute is now trying to enable. So hopefully we'll have this flywheel through which we can put a lot of different ingredients with great people and great ideas and come up with intellectual property and startups that are going to be world-changing, and also along the way be able to train a lot of great people. So if you are ever in Los Angeles or ever interested in joining us, please let me know. And this is again, we're very interested in a full speed ahead model, and this is actually one of the outside of one of our first buildings, and it's very nice area in Los Angeles in Westwood. Definitely come and visit us. And this building is actually won a lot of architectural design competitions. It goes in a couple of floors as well. So so definitely, I think this is what I wanted to kind of talk about most of the time. It's entrepreneurship is definitely something that is exciting, very time consuming, and there's a number of failures which I didn't talk about before as well, but it's a lot of learning process. So with that said, yeah, Mohsen and Houman, yeah, love to answer any questions.
Well, wonderful talk, Ali. As always, very informative and educational. I loved your talk and I learned a lot. As you know, we are also we have started a company on one of the technologies that we developed in my lab, and then we are going through this process as well. And then I saw that our CEO, who is again, he is an excellent a boss for us, is also among the attendees. So we learned a lot from you. So we will start. So if you have any questions, please you can raise your hand. We will start with those who have raised their hands. What I'm going to do is I'm going to unmute you one by one, and you can introduce yourself if you want, and then you can ask your questions. Please make sure that your questions are short. If you have multiple questions, then we can continue the discussion. We kind of want to have a kind of like a panel kind of discussion, more interactive session, so we can hear Ali's thoughts on your questions, and then and also your. So we will start with the first question first. So Atta, I unmuted you. Can you talk, Atta? Atta Ullah?
Please introduce yourself and your organization as well.
Atta, can you talk? All right. So let's go with Ashwini. So Ashwini Kumar, and I unmuted you. Can you talk now?
Hello, am I audible, sir?
Yes, we can hear you.
Yeah, thank you. That was a great talk. So my question is, since you developed a composite of gelatin and nanoclay for the embolization procedure, what was the degradation profile of this nanocomposite after the fibrosis of the site?
Yeah, so basically within a month, you get rid of about 90% of the material. So a lot of it is degraded, but it's degraded not just by itself, it's typically degraded by infiltration of cells. So that's really the mode of the degradation most of the time. So as cells come in, they kind of chew away some of these material, and then they deposit their own matrix. So that's kind of, and that's degradation time is kind of consistent with some of the things that we wanted with the cell infiltration and fibrotic tissue formation. So in that sense, it's ideal, and also with regulatory process, because as you start getting longer and longer degradation time, you really need to perform these experiments a lot longer and show that the material is not going to have long-term effects. So for us, that 1 month for majority of the material was desirable.
Right. Can I ask an extension of the question?
Sure.
Sir, so if that degradation depends on the infiltration of the cells, so that might also depend on the site of the procedure, because there might be different immune cells and non-immune cells at different sites. So that degradation might also change based on the site. Is it true?
I think that's true, and I think that's to some degree for us. Most of the sites that we're putting are going to be inside the blood vessel, so it will be somewhat similar. But if you're putting in something direct injection into subcutaneous tissue or some other portion, I'm sure there's going to be some differences in degradation as well.
Thank you very much, sir.
Thank you. So so the next question is from Mahdi Muradi. Can you introduce yourself and your institution, please?
Hi, this is Mahdi Muradi. Actually, I'm from a private company, Innonet Inc. from Canada. We have been able to develop a new technology based on the chitosan or chitosan nanoparticles. It shows very great in vitro and also in vivo efficacy. to inhibit all major microorganisms. It's also an organic material. We have been able actually to incorporate that nanoparticles into the different type of the polymers and also textiles to introduce the self-sanitizing characteristics to those biomaterials. I'm wondering if that kind of applications are somehow suitable for your institute in order to develop more in vivo studies on these new materials that we developed.
Yeah, and I think a lot of these types of things, there are a lot of synergies and the different types of biomaterials. So I definitely encourage you and everyone else who's got similar types of kind of systems to offline communicate with us, and we'll be very, very happy to kind of push things forward. I think that, and that's really where a lot of the exciting stuff comes in. Once people actually start putting in the right expertise together, it just I think would be fantastic. So I can already see a few different things about the chitosan nanoparticles that would make sense in our material. Wonderful, thank you.
Thank you, wonderful. So Rohin Shyam. Oops. I can't unmute you. Oh, there you go.
There. Can you hear me?
Yep.
Am I audible? All right. So very good morning to you guys. My name is Rohin. I'm from Vellore Institute of Technology, India. I'd already posted a question. My question was more related to the regulatory side. So in personalized medicine or tissue engineering, so let's say a 3D printed, bioprinted patient-specific cardiac patch, there will definitely be challenges from the regulatory side. So how does one overcome those specific, let's say, challenges from the regulations? How do you address?
Well, I think the first thing is to really try to understand that the regulations are at the end are going to be useful for protection of people, but also of the company, right? So because if there are adverse issues, then no one wants that, right? So a lot of but at the same time, you want to kind of make sure that you're doing all the safety and efficacy type things that are required without breaking the bank, right? So at the end of the day, you need to still pay the bills and push things forward. So I think there is some kind of a balance that you want to do all the important things that are required and really shows the safety and efficacy. At the same time, if there's pathways that you can do that without having to spend $30 million, then that could be good. So what we've been working at is to really look at the predicate devices. Are there other things like this that have similar types of characteristics that we can use and justify that, okay, it's going to be just as kind of safe and efficacious as those other things. So that's one of the ways I think to deal with regulations. I think there are huge differences between something that really needs a clinical trial of a significant degree and other things that are can potentially have a smaller clinical trial based on some predicate devices. So I think that's kind of been our approach to kind of look at what are the different ways in which we can ensure that things are going to be safe and efficacious, and at the same time make sure that we're taking the pathway that's going to allow the company to succeed.
Yeah, okay, cool. Thanks. Just an extension to that question, if you don't mind. It's the same. Like, let's say when you're talking about patient-specific part, you can't go back and forth trying to get an FDA approval for a patient-specific patch,
Yeah. Yeah, no, and I think some of those very specific things like 3D printed. Yeah. So are you guys good?
Yeah.
Mohsen and Hossein, you guys are saying something in the background. Am I working okay? Am I okay? Yeah. Can everyone hear me? I guess yeah. That's because I can hear you guys.
Please continue. Yeah.
We can't hear Mohsen now.
Okay. But what I was going to say is that with very specific things that need to be tailored every time to a patient, I think there's still a lot of questions about what's the best way to get approval and things like 3D printed medical devices or 3D printed things which need to be tailored every time. I think for those, there's still some work that the FDA has to do with some of the companies to really create the right process for it.
Okay. Thank you very much for answering my question.
Well, can you hear me now?
Now
Yes.
I can hear you.
Wonderful. So let's
You
go
were
on.
waving up and down. I got scared. Yeah.
It's like, yeah. Well, we have questions in the question box as well, which I wanted to ask. So we have Dr. Pepas, who have asked a question about how do you know that an idea can be commercialized?
Wow. So first of all, just getting asked by Dr. Pepas is a big honor. Dr. Pepas, as I mentioned during the talk, is one of the leading pioneers in the area of biomaterials. So with my humble opinion, I can try to answer that question. I think when it comes to translation, I think some of the things that are important is to try to understand if a particular area is actually an area that needs to have major innovation in it, and that major innovation is actually going to change the standard of care. There are areas that are, so when I interact with clinicians, a lot of times clinicians come up with very specific problems that are very tailored to their procedure that's only done, let's say, a couple hundred times a year in the entire US. And even though that's an important problem for them, it's not necessarily going to be a good basis of a startup company. So I think for me, it's been, and that's been a learning experience, because there's been times that I spent a lot of time on something like that, and at the end I realized, okay, I maybe shouldn't have started. So I think really understanding the patient need and how big that need is, and how many people it affects, I think is important. And then second thing is how well the idea is. What I like to call is the 10x. How's what you're doing makes that state of the art 10 times better? Because if it's a 10% better, then sometimes even if everything makes sense, but it's just not well differentiated enough from the existing state of the art to kind of justify a lot of investment. So if what is the 10x and how important is the problem? And I think if some of those things start kind of making sense, then I would then try to kind of push it through. And again, I'm sure Dr. Pepas knows a lot more about all of these. So but that's my humble opinion.
Well, thank you very much, Ali, and then thank you very much, Dr. Pepas, for asking the question and also participating in this talk. So we have another question from Dr. Woo-Soo Kim from Simon Fraser University in Vancouver. So the question is about a method of encouraging graduate students or postdocs to the direction of entrepreneurship. What is your magic? It's sometimes difficult to convince or persuade the grad students to follow an entrepreneurship path. So what do you do to encourage them? Yeah. I'm sure you have a lot to say.
Well, for me, I'll tell you my own story, right? So I definitely became more and more interested in entrepreneurship as I learned more about how people have used it to make impact, right? Because again, as scientists, researchers, we're trying to maximize our impact. And the reality of it is that if you're exposed to a lot of examples where people have used entrepreneurship in making huge impact, then you become excited about it, right? So that's really, I think, what separates a place like MIT, let's say, and working in someone like Bob Langer's lab from working in some other place, right? Because you get exposed to it over and over and over again until it kind of gets into your DNA. Even if it wasn't there at the beginning, you definitely get some gene knock-in into your DNA because of seeing it and hearing it and seeing how it's made major improvements in people's lives, right? So I think that's really important. Just kind of programs that not just tell you the nuts and bolts of what it takes, but inspire people to go into that direction.
Wonderful, wonderful. Thank you, Ali. So we have received plenty of questions. I don't know how much, because we were supposed to finish this basically talk at 10, but I don't know how much time do you have, Ali?
I have time. I'd be happy to answer more questions. Yep.
Wonderful, wonderful. So let's go to the to those who raised their hands. We have Nihal. Let me just unmute you, Nihal.
Hello, Nihal Engin Burana, Sparta Medical. Ali, what I would like to ask is, in the Terasaki Institute, up to now, what you have done is more top-down, in the sense that having a experienced CEO. Do you plan to do more bottom-up, like enabling the postdocs to form their own companies, moving in that direction?
Yeah, well, I think you're totally right. One of the things we're trying to do is to really get the people who want to make these things happen, right? Because top down only allows one person's kind of thoughts to get it incorporated, right? So my experience from the time I was in Bob Langer's lab is that it's good for me to be the gatekeeper, but allow lots of ideas to kind of develop behind the gates and see which ones of them kind of meet the bar for us to kind of reveal to the world. So that's really what we hope to do at the Institute as well. So really be able to get top-notch people and then give them freedom. Give them freedom to come up with ideas, give them freedom to explore with other people, to interact with other expertise or institutions, so that we can actually come up with the best ideas and come up with the best solutions to them. So, you know, and one of the programs that we have to kind of encourage that is actually something called the Terasaki Fellow Program, where we bring in people who we think are at very junior stage. Let's say they just finished their PhD or a year or so into their postdoc, but we bring them in with the intention that, okay, we're gonna give you some flexibility and freedom to come up with processes or ideas or thoughts that you think are gonna be world-changing. And we give you that freedom to kind of explore and try to make it happen. And we provide resources as needed to enable people to do that. So that's, again, and I think that works for both people who are interested in entrepreneurship, so you come up with ideas that you can actually spin out, or people who are interested in academia. So again, you come up with thoughts about what is the next big thing and try to enable it, and then become an independent investigator, ideally even at the Institute.
Thanks.
Awesome. So the next question is from Mehnush. Mehnush, can you talk? We can't hear you. No. Let me just unmute her again. No, your microphone is not working. Maybe you can write your question in the question box. So, so we have Venkata, if I pronounce your name properly. You also raise your hand.
Yeah, I am a PhD student from McGill University. So I read many papers from Professor Khademhosseini, and especially in 3D bioprinting part, I mean, like, he didn't explicitly focus on laser-based bioprinting. May I know what are the reasons for that? I mean, like, are you pessimistic about that approach or anything like that? Femtosecond laser-based?
Yeah, so, you know, so one of the things I always say is that I don't really, I'm agnostic to a lot of different technologies, because I think at the end, different technologies are good for different things. There are some limitations with any of these different technologies, right? So whether they're extrusion based or kind of stereolithographic or other things. So I definitely think there, for each one of these, there's areas where there's opportunities to do. So you know, one of the things, if I haven't done something, is because, you know, I definitely wished I could do it, except, you know, I haven't had the bandwidth. but I definitely try. Yeah, I don't see any limits on what 10 figures can do. So I always tell my students that if you get inspired by people like Dr. Pepis and Dr. Langer, I think you realize what individuals can accomplish.
Right. Thank you very much. Yeah.
Okay. All right. So so let's look at the question box. We have wow, so many questions. Yasaman Pushidani from Tehran Polytechnic. Her question is, what is the best way to know the market needs? How we can find a potential field that we know can be commercialized?
Yeah, so I think the market need, first of all, it's good to understand the clinical problems, at least in our area. Just, there are big problems out there, like, there's cancer and cardiovascular and, particular things, diabetes. There are big problems out there, right? So once you understand the big problems, then you actually look into them deeper and you start seeing, okay, what is the actual things that have been done and what are the specific problems that remain in those particular solutions that have been implemented. So this is a process that's, just kind of, there's many layers to it. There's, initially you would, kind of do some survey of different areas and then you start seeing some opportunity, then you zoom in and dive deeper into particular areas. But, the main thing is to really read broadly, be open to many different types of things, be open to ideas, be open to areas, and then interact as much as possible with people from different fields, and try to not be limited to your own kind of direct, immediate expertise, but try to be just open-minded. What I had to go through in my own development is, I initially developed my own expertise in a particular area, but then every time I did something, I added something to it that I didn't know before. And that's how you can start kind of building in some core knowledge in a wider area.
All right, wonderful. Thank you, Ali. We have another question from Ava, who is an electrical engineer. She's a master's student. And then the question that she asks is that whether an electrical engineer can work in your area, Ali. Then
yeah, good question. So I would say, again, the way I think about all of this is get the best people, independent of their background, independent of where they're from or anything. Because the best people can pick up things and can apply their background to different areas. So in our institute, we have number of electrical engineers who are working on different tools and technologies. So as I mentioned, one of the areas that we're very much interested in is to create wearable devices or implantable devices that can sense and deliver compounds. So that's one area. There's a lot of data science that we're interested in incorporating into these different approaches. So I think there's definitely a lot of opportunity, not just for electrical engineering, but I think even beyond and beyond, even philosophy or things like that, people, we have a big initiative at the Institute that we're starting to develop on art and science. So all of these fields, I think, can benefit from each other, and just being open to it and seeing opportunities, I think, is the way to do it.
Awesome. Great news for many researchers from different fields. So let's go to those who raised their hand. Mehnush, I think your microphone should work now.
Yes, it works. Can you hear my voice?
Yes.
Hello, guys. Thanks, Dr. Khademhosseini, for your interesting talk. I am PhD of mechanical engineering, and from mechanical engineering aspects, when I listened to your talk, several mechanical properties is so important for consideration, but I saw that just stress, yield stress, and strain has been considered with you. But as you know, these nanocomposites is under loading, dynamic loading, and fatigue life is so important, or another mechanical properties such as swelling, such as shear stress, friction coefficient, and wear is so important. Have you measured them in your research?
So yeah, it's definitely a great question. So we have looked at some of these because the ones that we think are interesting for some of the properties of the material. So for example, as you mentioned, some of the things like creep, for example, is important. What some of the things related to adhesiveness of the material to the tissues is important. And so I think there's definitely a whole set of tools that technologies that people feel for this class of materials, the embolic that are kind of material gel-based embolics that are kind of characterized and people use, and even for regulatory, it's important. So we've been kind of mostly sticking with those standard approaches that have been kind of previously published.
And have you modeled this inside the body? And have you measured these mechanical properties? Because modeling is so important, and interaction between these and tissue. So
definitely, I totally agree. And that's maybe an area of collaboration. So I think there's definitely very important kind of modeling type work that is needed as well. So we've done some modeling, but mostly fluid dynamic modeling of looking at inside the blood vessels and shear stresses that these kind of materials get exposed to, but not necessarily the mechanical properties of the material model.
My expertise is atomic force microscope, and I have worked for several polymers and nanopolymers, but not exactly as biomedical tissue. And if you are interested, I'm so interested in collaboration. I will contact you for research.
Sounds great.
Thank you. Thank you.
Thank you. So we have Elham Salimi. Can you please introduce yourself, your institution as well?
Yes, my name is Elham Salimi. I'm an assistant professor at the University of Manitoba, just recently started. And thank you, it was a very interesting talk. I really enjoyed it. I actually want to ask, actually seek an advice from you on how to deal with the interdisciplinarity nature of this kind of work and the communication between the people of different backgrounds. Like, I personally find it a little difficult. I have a PhD in engineering, in electrical engineering, and I find it sometimes hard when talking with doctors and medical people, like as if the impression I get from them is that you engineers are intruding into our field. Go back to your own things. So what kind of advice do you have for like mitigating this kind of interactions?
Yeah, well, I guess the first thing I would say is that you can't make everyone happy. So there are people that you should just kind of try to avoid, and situations that you should try to avoid. So that's the first thing. I just think if there's someone who's not ready for being a positive, interactive collaborator, you can't force them into it. The other thing is, I think if you try to learn some of the basics of some of these different areas, and at least through different ways, you can do a little bit of your homework. So when they talk about particular terminology, at least some things about it, right? Some of that comes with experience as you kind of start doing things in a particular area. Let's say if you're doing more biomedical stuff, then over time you start picking up more of the terminology, more of the way different people kind of work. And obviously you need to have a lot of mutual respect. So if that mutual respect is not there, then I would say you shouldn't be trying to force that interaction with those people. And really down the road, you don't really need too many collaborators. You just need some good ones. And that is all you need. So that's why, for me, when I look at my own past 15 years and my collaborations, there's been just a handful of people that I collaborate, not hundreds of people, right? So again, maybe I've published with hundreds of people, but the collaborators that I've really been interacting with just are a handful. So I think, that's what I would encourage you to do, to find the right collaborators who are interested in, pushing things at the interface between two different fields, and try to kind of go from there. But what you're mentioning, I think, is definitely important, and something that it's more accepted to collaborate between fields and places like, let's say a place like Boston was easier compared to a place that's not as much used to it, like, let's say, some other places in the United States.
Thank you.
Thank you. So we have received so many questions. Again, Ali, whenever, you think it's...
Yeah, so maybe what we can do, we can go another 6 minutes, and then we'll go from there.
So what I'm going to do is I'm going to ask one question from the question box and one from those who raise their hands. And I have a list of questions myself here, which I think I'm going to call you later and then ask you. I didn't even get a chance to ask my questions, Ali. So we have Ankit from IIT Roorkee. The question is about your advice for early career researchers wishing to enter the field of translational research in bioengineering.
I think one is you have to kind of be willing to understand there's going to be lots of ups and downs, and understand that you have to have the ability to persevere through all of these challenges, and that it's really a long-term endeavor. There's very few short-term super successes that one can kind of say, yeah, I started. Most people have to go through lots of difficulties, but at the same time, one has to maintain their passion and continue to improve on daily basis, right? So one of the things, in fact, I encourage people to probably check out is on the Terasaki website. I've kind of set a series of like leadership principles, right, which are kind of advice or things that people can kind of adhere to, kind of rules of success. So I encourage people to look into it, because like these are, and I have them as such an important part of my life that I have it right on my desk on my thing. So basically, be opportunistic, move fast and get things done, have a backbone, disagree, but commit, dive deep, act with integrity and earn trust, persevere, take ownership, be frugal, try to get the most things done with the least resources, increase standards and raise the bar daily, learn and be curious, hire and develop the best, care about patient needs. So those are like, just general things that I think, these kind of leadership principles that I think if one is trying to get into that world, is a way of living that I think, one can start adopting.
Awesome, awesome, Ali. Just one last question from those who raised their hand. I apologize from everyone who didn't have a chance to ask their questions. We have limited time, and Ali is pretty busy. We were supposed to have this meeting for 1 hour, but now we're half an hour over time. So again, I mean, we will post this the recorded video on YouTube, so you can subscribe in our channel, and then you can ask your questions in the comments there, and then hopefully Ali will have us to go and then take a look and answer your questions.
Let me give you a plug. Anyone who wants to ask me questions, I have my fireside chat series. You should come.
There you go.
I'll give you a lot of.
There you go. Yeah, fantastic. So the last question is Bianmei Chao, probably. Sorry if I didn't pronounce your name properly. So Bianmei, can you connect your microphone? Can you unmute it? I unmuted yours. Can you talk and then see if... Let me unmute you one more time. Okay. So then if so Bianmei. Okay. So let's go with Norbert. Norbert Radasi.
Hi, hello. Hi, Ali. Can you hear me?
Yes.
Oh, good. Thank you very much for your time. I really appreciate it. And I wanted to ask you about your recent paper where you envisioned this intracorporeal tissue engineering. And I really like the idea of one day having robotic assisted minimally invasive surgeries. And actually, I'm an assistant professor at the University of Edinburgh working very closely with surgeons. So I showed your paper to one of my good friends who is a kidney surgeon, and we wanted to ask you if you think one day we can use it for removing cancer from the kidney without him cutting the patient open, and whether you think it would be possible to commercialize it.
Yeah, no, those are great questions. You know, so I think definitely there's a lot of opportunity in kind of integrating robotics into kind of really advanced futuristic surgical things. I know there's a lot of surgical robotic type systems now, but I think integrating techniques like biomaterials delivery or you know, kind of being able to use kind of like printing technology that I mentioned, or advanced ways where you can, let's say, take a biopsy or remove, you know, do a full resection of the tumor, but also at the same time be able to deposit, let's say, anti-tumor agent or a drug delivery platform. So I think, you know, this kind of integrative approaches where it's not just a one system platform, I think is going to be the way of the future, where you would have the advanced robotics combined with the personalized cells, combined with the personalized materials, in a way that can actually best address the problem. So regarding the specific problem that you mentioned, I can definitely see, you know, in a way in which through kind of non-invasive or minimally invasive imaging, that procedure can be accomplished, and then after that, you know, there could be a delivery of cells and materials and drugs in a way that it would prevent the recurrence of these things, all in a way that's kind of automated or robotic based.
Okay, thank you very much, and I think it's an excellent, very interesting future. So thank you, Ali.
Thank you.
Well, thank you very much, Ali, and for your again inspirational talk. I personally learned a lot from you. As always, whenever I listen to your talks, I mean, I learned something new. I had actually, I wanted to say that I had a privilege to work on partly on this the shear thinning material with Rami and Reggie, and then I remember that we didn't sleep for 36 hours to collect some data. And I remember that I went to Mass General Hospital with Rami in his control room to see how he, plumbs when he was actually plumbing patients, and it was very nice when I was in there. Thank God I didn't go into the operation room though. But thank you again, Ali, for your time, for your excellent talk. And,
yeah, thank you guys, and thank you for this incredible initiative that you started. I think a lot of people will get a lot out of it, and I'm hoping that the Terasaki Institute will be a big partner of you guys going forward.
Well, for sure. And then we need to chat about this maybe later. But thank you. And then before we end this meeting, I just wanted to announce that our next speaker is Dr. Azadeh Guderzi, CEO and co-founder of Pomegranation Solutions Institute. She will talk about how to upgrade your toolbox with financing, marketing, and mindfulness. So I encourage you to basically attend her talk. Those who already registered for this talk will be automatically, their seats will be reserved. Please spread the word. We have 800 registered for registrations for this talk, and we hope that this program and e-seminar series will continue. We will have speakers lined up for the rest of the year until the end of the year. So please make sure that you follow us on Twitter, LinkedIn, Facebook, pretty much every social media that you can name, and then we will update you with the news about the speakers. And if you have any questions about this talk or the future talks, please also feel free to email me and Human. Our email was provided in the introduction slides. So thank you very much, everyone, again for participating in this talk. Thank you again, Ali. Also, we would like to thank Revolution again for sponsoring this this event. And I let you go. Have a good day.